say never let monkeys eat bananas is why. So this is a much better diagram. I just wanted to touch on it really quick. These are the 5 players in the leukocyte family, right? Neutrophils, eosinophils, basophils are what? Granulocytes, and these guys, lymphocytes and monocytes, are agranulocytes. Even though I told you when you look under the microscope you're going to think I'm a liar, because every once in a while you'll see granules in your lymphocytes, but they are agranular cells. So, the colors, much better picture in your book now. But the granules, pinky purple, and those nuclei that are weird, multi-lobed nuclei, what do we call that? Big long word. Polymorphonuclear leukocyte. This is a better looking eosinophil than the other crappy slide I put up there before. And you see that the granules actually look like little balls now instead of sand, and they're orange. For the eosinophil, basophil, they're balls and they're black. That's how I remember it. Lymphocytes, we don't have that weird nucleus anymore. We have a nice round nucleus. And that, what was the other characteristic that we pointed out? Yeah, the blue cytoplasm. And then the monocyte is more on the grayish side and much airier looking nucleus. This one happens to be lobe shaped, but sometimes they can be round. You want to look at how compact they are. Lymphocyte, much more compact and dense. Monocyte, much more airy. Also, the edges of the cell are kind of fluffy too. Don't forget your progressions. What was that? No, it's platelet formation. Ooh, you adding another exciting word there. Remember, platelets are other formed elements that we find in the circulatory system, but they're fragments of a cell. Big, huge cell called the megakaryocyte. When it goes to squeeze into the circulatory system, it's going to blow up. This is hemostasis. Very good. And all those little factors. Don't forget, what do you have to know of this diagram? Everything here, this common pathway and the difference between intrinsic and extrinsic. Yes? So know these. And then the formation of, at the bottom here, prothrombin activator. Yes? That's a nice clot with blood cells. So, that brings us to thrombolytic disorders, which we touched on at the end of class. Problems with platelets. If I don't have enough platelets or they don't function properly, I can have problems clotting. And just a basic definition of the disorders described for you on page 650 and 651 would make me a very happy camper. So thrombi and emboli, know the difference between the two. What is the difference? One stationary, which one? The thrombus. Thrombi are stationary, emboli move around, and they're both clots. Anticoagulant drugs are given to patients who spontaneously form these clots for some reason. Or they might have disorders that might lead to spontaneous clot formation. Thrombocytopenia, what's that? Not enough platelets. That's a problem, right? Hemophilia and disseminated intravascular coagulation. Again, forming clots when I really don't need them is a problem. So the next thing we go into, in a kind of confusing concept with respect to blood, is something called the ABO blood group antigens and antibodies. First of all, what I want to do, looking at this chart, is I want you to understand the definition of the two. An antigen is a substance that can elicit an immune response. What the heck does that mean? It activates an immune response. What does that mean? An antigen can activate an immune response. Yep. It has the ability to trigger a series of reactions that will alert cells of the immune system that it might be foreign. The immune response will include the production of chemicals to neutralize that foreign substance. And those chemicals are called what? Antibodies. They'll both start with anti, so sometimes we get confused, but antigens generate immune responses. Very good. So antigens are going to cause cells to make antibodies, chemicals to neutralize them. Now, not all antigens are foreign. If I look at any given cell in your body, you have tons of antigens, these proteins or carbohydrates or combinations of the two substances on the surface of your cells. They are your cell markers. Many of them have functions for the cell. They might let things in and let things out. But those are your antigens. Your cells have antigens that define them as yours. We call them self-antigens. Would I elicit an immune response against myself? Under normal circumstances, no. There are groups of diseases where our body gets confused. The cells get confused. And they actually start attacking self. Does anybody know what those diseases are called? Autoimmune diseases. Those are not normal. So we're talking under normal circumstances, yes? So your cells have very specific markers on them. Again, those markers perform functions, but some of them are just markers. Like, hey, this is who I am. And who are they for? Who takes care of keeping you safe in the body? We've talked about them in this chapter. Yeah, those white blood cells. Remember I said the thymus was T-cell kindergarten? Well, guess what the T-cells go to learn in thymus kindergarten? They learn to recognize self-antigens. If they don't pass their final exam, it's a tough world in my human body. If they can't recognize self and they attach themselves too tightly to self-antigens, that's failing the test. Guess what happens to them? They're induced into self-destruction because we don't want them around. If they can't recognize self-antigens, we don't want them floating around my circulatory system. We don't want them hanging out in my lymphatic system. And we'll talk about these guys again when we talk about an immune response in the lymphatic system. So the reason I gave you that little up front about antigens is because when we talk about blood, there's very important antigens on the surface of blood cells that are going to help with many different things in the body. These antigens on the surface of red blood cells are called our ABO antigen group. Now that sounds familiar to you, doesn't it? Like what type blood are you? Do you know? You're O. I'm A+. What's that mean? What does that mean? What's your blood type? Well, basically your blood type tells us a little bit about what's floating around in your circulatory system. The first thing it tells us is what type of antigens you have on the surface of your cells. So if you are type A or B or AB or O, I know just by you saying that what antigens you have on the surface of your cells. So if you look at this chart, I want to first look at the antigens. So the antigens are substances on the surface of the cell. And if you're type AB, you have two different antigens on the surface of your cells, type A and type B. If you're type B, what kind of antigens do you have on the surface of your cells? B. If you're type A, what kind of antigens do you have on the surface of your cell? A. And if you're type O, you don't have any A and you don't have any B. Do you have antigens on the surface of your cells? Yes, but you don't have any A's and you don't have any B's. Yes? So that ABO is just a very tight little column of antigens that we're talking about, basically A and B. The reason they're so important is because when we talk about transfusion and blood replacement, which they talk about here, has anybody ever had a transfusion or know somebody who's had a transfusion? I can share my blood with you. The beauty is it can help you through some tough times. If you have tough times with volume, say you have a bleeding disorder, or you get into an accident, you lose a lot of blood. I can donate my blood and you can use it. But I have to be very careful because of these antigens and the other guy's antibodies. Normally in your body, the only time you're going to make an antibody is when a foreign antigen comes in. So I'll give you an example. Anybody have a cold right now? Okay, what's it probably from? Some nasty, rotten little virus, right? Virus gets into your body, right? Foreign antigen, and alerts your cells that it's there. Your cells will then go through an immune response and make the chemicals I just mentioned, antibodies, against that foreign substance. And eventually, and you even mentioned it to me, you start to feel better. Why do you start to feel better? Yeah, those antibodies are going to start neutralizing those antigens, and the cells of the immune system are going to get rid of that crappy, nasty little foreign invader for you, and you start to feel better. That's under normal circumstances. But for some reason, and we still don't know why, you will make antibodies for the ABO blood group antigens, and it's only in this case, just ABO. You're going to make antibodies against the antigen that you don't have. We still don't know why, but you do. So, for example, if I'm type B, which antigen don't I have? I don't have A. My body will make something we call naturally occurring antibodies against the antigen I don't have. So in this same diagram, we also see these little purple triangles. Those are anti-A antibodies. So when I look at my blood on the whole, I have A cells, and in my blood plasma, I have B cells. I have B cells, and then I have anti-A antibodies floating around in my blood plasma. Yes?
Is that why it's like a problem if you're punching your blood type?
No, that's another antigen we're going to talk about in a minute. Okay? So, if I'm type AB, which one don't I have? I have both. I have A and B. So what type of antibodies do I make? Neither of those. Because that wouldn't be beneficial to me, would it? I'd attack my own cells. So if I'm type B, I have B antigens on the surface of my cells and anti-A antibodies in my blood plasma. If I'm type A, I have A antigens on the surface of my cells and anti-B antibodies in my plasma. And how about type O?
You don't have any
Correct. So I don't have any antigens on the surface of my cells. I don't have A and I don't have B. But remember, I'm going to make naturally occurring antibodies against the ones I don't have. So I have both anti-A and anti-B antibodies in my plasma. Now you've seen this word before, right? Universal donor. Be very careful with that word. Yes? So if I'm bleeding out on the street and I'm type O, I can take anybody's blood here. Is that the truth? No. Because if I give type O blood, whole blood from a type B patient, what's going to happen? My antibodies are going to attack those foreign cells and cause what to happen? A rejection. Clots. Antigen-antibody reaction creates clumps. These clumps now can be recognized by other cells in an immune response, phagocytes, and they come along and they grab the clumps and they do their thing. But if I have a whole bag of clumps that I've now added to my circulatory system, could that be a problem? Yes. When you get blood in the hospital, if you've ever seen a patient get blood in the hospital, are they getting whole blood? Plasma and cells and all of that? Very rarely. Rarely, right? What are they getting? What's in that bag? Just red blood cells. They're called packed red cells. So what happens when you donate blood? Most of the time, what happens to your blood? They're going to separate it. They're going to wash off all your red cells because they don't want any residual antibodies left. And they're going to put them in saline, a nice isotonic solution, in a bag to donate. Because really, when somebody needs blood, most of the time, they need the oxygen carrying capacity of your red blood cells. The other components I can use too. So you might just get FFP. What's that? Fresh frozen plasma. Yes? Because maybe you only need stuff in the plasma. Or maybe you only need some volume. Okay? Maybe if you're a hemophiliac, you might need some of those factors you might be deficient in, and that's all you need. You don't need the cells. So we separate them, and very rarely does someone receive whole blood. The reason very rarely they get whole blood is because we'd be adding way too much volume to their circulatory system. So if I only need the cells, then that's what I'm going to give you. So can I give O cells to anybody? Yes. Because why? There's no antigens there for my antibodies if I'm type A, B, B or A to react with anything, right? But if I'm type A and I give my cells to type O, very bad. Yes? If I'm type B and I give my cells to A, very bad. Yes? AB, he's kind of seeing good things too, right? How come? He doesn't have any antibodies. So if type B gives him blood, we're good. And if we're type A and we give him blood, yeah, we're fine. And O, only what? Only O. Only O can get O.
So if you're dying, you want to be
Yeah. Yeah, it's common. O is common. Now, the ABO antigens aren't the only ones we have to worry about. Remember, they aren't the only antigens on surface of cells. There's also a very popular one that's also part of your blood type called RH. Well, where does it, well, if I say A positive, that's the RH. So, RH is another antigen. It's called D. And if I have it, I'm positive. If I don't have it, I'm negative. This antigen follows the other antigen rules I mentioned before, these ABO guys, who are kind of the exception to the rule. The only time I'm going to make antibodies against the Rh antigen is when? No, when am I going to make them if I don't have them? No. Yeah, only if for some reason that antigen gets into my body. So here's the problem with mother and baby. No matter how hard you try to separate mother and baby's blood during the delivery process, no matter how smooth it goes, the baby's blood is going to get into your system, even if you have a C-section. Yes? So if you are an Rh negative mother and your baby happens to be an Rh positive baby, what are you going to do when that happens? No, not yet. But that blood's going to get into your system. Your system is going to recognize it as what? Foreign, and you are going to elicit an immune response. You're going to cause cells to make antibodies that you didn't have before against an antigen, that foreign Rh antigen. And that's okay. Everything goes smooth, first birth. Not a problem. But now, say you get pregnant again. And your baby is Rh positive again. Well, guess what? You kind of share the circulatory system during the child's stay. And you particularly share plasma and the fluids that float through plasma through the placenta. And unfortunately, anti-D or Rh antibodies are small enough that they're going to pass into baby's circulatory system during your sharing process. Do you see where the problem's going to start to come in? So as baby's blood starts to form, if that antigen comes in contact with the antibody that passes through your placenta into their body, it's going to cause what? Immune response, clots to form. And that's what they talk about. Or they talk about it in, well, maybe they don't. But that's why it's important to know your blood type when you're pregnant and your mate's blood type. And even if you're an Rh negative mother, it doesn't even matter what your mate's blood type is, because we're going to take precautions against that. The way I can prevent the body from going through an immune response is to give it all of the antibodies it needs to fight off a foreign antigen. And if I do that, the body won't ever go through that major immune response. So any Rh negative mothers in the group? No? No? Yes? What'd you have to have around that birthing time? It gave you a shot in your butt. Do you know what it was? It's called Rogam. And do you know what it is? It's a big dose of anti-Rh antibodies. So any blood that child passes to you, if there's any chance that there is that antigen present, that big dose of those antibodies will neutralize it and your body won't go through an immune response. So the next time you get pregnant, you won't have those antibodies circulating in your system because you never will have made them. We'll talk about that when we talk about innate and adaptive immunity in the immune system chapter. Did you have a question? Or are you just going to tell me what it was? Rogam. Okay, so that's why you have to have that if you're an Rh negative person. Does everybody understand then why someone might undergo a transfusion reaction? And that reaction can be very small or it can be very large. When you donate blood, it gets separated and packaged. And one of the things that happens is there's a big long tube where there's some blood in there, right? And you ever notice that they kind of leave it in there, and sometimes they'll do it right on site. They make little crimps in it. What they've created is little tubes of blood for themselves. And when you donate blood, and your blood gets packaged, those little tubes are going to go along with your blood to the blood bank. When a patient needs blood, one of the first things that has to be done before they get their blood is we have to do a blood test. We have to take a tube of blood and bring it down to the blood bank. What do you think they're going to do? First, they're going to type your blood, see what type you are. So then they can go get a bag from the fridge. So if you're type A positive, they're going to go get a bag of what from the fridge? A positive. The next thing they have to do is make sure it's not just A's and B's and RH's that they're worried about, but if you have any other antibodies that you've created against blood cells. And that can happen. So they do a cross match. So they take that tiny little tube of blood and they separate it, the cells and the plasma, and they do what with it? In a tube, they cross it with your blood. So they take the cells from the donor and put your plasma on the cells. And they take the plasma from the donor and they put it on your cells. And then they look under the microscope to see what? If there's clumps that form, am I going to give them that package of blood? No. So that's what a type and cross match is. So those antibodies circulating in your circulatory system can be a big problem with respect to blood donation. Also, when we talk about tissue donation, there's even more antigens that we have to worry about. We'll talk about those guys when we talk about MHCs. They're called major histocompatibility antigens. There's two groups of those guys that we'll talk about when we talk about immune system as well. So, understand blood typing. If I want to type my blood, see what type of blood I am, and we'll hopefully do a little bit of this in lab, we're going to use the antigen-antibody concept. So I can make antibodies against foreign antigens in any animal. So if I take my blood cells, say I have type AB blood cells, and I inject them into an unsuspecting cute little goat, what is the goat going to do? Oh, that's a good one. I walked right into that. That's bad. That was wide open. What's the goat going to do besides say that's bad? Any ideas? Yeah, their body's going to undergo an immune response. They're going to kick in cells from the lymphatic system, those lymphocytes we talked about, and we'll talk about more. And eventually they're going to produce a whole bunch of what? Antibodies against my AB cells. Now what can I do? I could go get them. In the lab, what can I do to the goat now? And I don't even have to kill it to do this, which is nice. That's why we pick on goats when we make stuff like this. It's floating around in the goat's blood plasma. That's a hint. Yeah, draw blood from the goat, separate out the plasma, use some techniques to separate out the specific antibodies that the goat made, and then package them in a little bottle. It's called a reagent. And there's different ones. Anti-A, anti-B, and what's the other one we're concerned with? The RH, anti-D. So if I have these little vials now of these specific antibodies against the specific antigens, I can take the chemicals and put them on my cells and wait for a reaction. So if I'm type AB blood, and I put anti-A antibodies and anti-B antibodies on them, what's going to happen? Big, huge clumps. That's what happens in your body if antibody and antigen react. So if I'm type AB and I put the chemicals on, both of them will clump. Because why? I have both of the antigens on the surface of my cells, A and B. If I'm type A, only A is going to clump. because I have A antigens on the surface of my cells, and the anti-A antibody will cause clumps. What happens to B? Nothing. I don't have any B antigens on the surface of my cells, so the antibody's not going to react with anything. If I'm type B, you getting the point? So if I'm type B, nothing happens with anti-A antibodies, but when I put anti-B antibodies on, we'll get big clumps. How about type O? Nothing. How come? Because I don't have either of them. I don't have A and I don't have B antigens on the surface of my cells.
You have antibodies.
I have the antibodies, so of course it definitely wouldn't react, right? Say it again, hon.
When you have the RH reaction, is it the same?
Yep. It's not going to be as violent, believe it or not. So we'll get a reaction, but it's going to take a little bit longer, and it's not going to be big, huge clumps. It's going to be smaller, more fine clumps. And hopefully, we'll hopefully get a demonstration of that from your lab instructor in lab when we do blood next week. Okay? So that's blood typing, and that's what they'll do in the lab. They talk a little bit about diagnostic blood testing, and one of the things you're going to do in your lab class is something called a complete blood count. Well, part of it, we're going to do the differential white blood cell count. A complete blood count, a CBC, counts all of the different components. So it'll send it through a little machine that will count the number of red cells per cubic millimeter and the number of white cells per cubic millimeter and the platelets, and they'll do all kinds of little calculations on the shape of your cells. And then that little smear that you're going to actually do in lab will differentiate those white blood cells. Now, back in the day, the machines couldn't tell the difference between white blood cells. Now, fancy schmancy machines can actually give you an estimate of the different types of cells. All right, get your clickers out. I think we already did that one, right? Infected plasma, we did that one. And red blood cells, efficient oxygen. Did we do that one? Okay, let's start with this one. So, red blood cells are efficient oxygen transport cells. Of the following characteristics, which is the major contributor to significant oxygen carrying capacity of a red blood cell? Yep. Oh, A, B, C, D. Sorry. Hopefully A, B, C, D. Got put the letters in. Let's see how many get it right. 5, 4, 3. Just click. Remember, it's for attendance. 2, 2, 1. What's the answer? Very good. Do red blood cells contain myoglobin? What's myoglobin? Yeah, we talked about it when we talked muscles. It's in muscle, not red blood cells. Let's see. Next, each hemoglobin can transport how many oxygen atoms? Five, 4, 3, 2, 1. What's the answer? 4. Well, let me see what I got. 4,000? We're only talking about one lousy hemoglobin molecule. The answer is A. It would be awesome. Oxygen binds to the what portion of hemoglobin? Go with your gut on this one. Five, 4, 3, 2, 2.5. Did everybody click? 35 our number? What's the answer? Iron. Well, globin is what? Globin is protein. Oxyhemoglobin, what is that? That's oxygen attached to hemoglobin. Oxygen binds to heme. What's smack dab in the middle of heme? Iron. That's where I clicked B, I'm sorry. Well, that's not right. It doesn't matter, I'm not grading them anyway, but C is the answer. Amino acid, that's part of a protein, right? A patient with low iron levels would experience which of the following symptoms? Now remember, the question says, which of the following symptoms? You can eliminate, that's all I'm saying. 5, 4, 3. What happened to 35? Everybody click again, just in case. 35. All right, no 35. What's the answer? The answer is C. You can't experience a decrease. That's not a symptom. All right? The decrease in white blood cell count is not a symptom. Can I just say, oh, I feel a decrease in white blood cell count? What is the symptom? So you can eliminate 2 things right off the bat. Yes? Although I do use the excuse for going out for ice cream that my calcium levels seem to feel very low, but that's a lie. All right. A hematopoietic stem cell will give rise to Five, 4, 3. You thought you had pressure on the online exam. You know, me yelling at you. 5, 4, 3, 2, 1. What's the answer? D, all of the above. All of those guys. So erythrocytes, I'll turn into erythrocyte if I come in contact with what? Erythropoietin. I will turn into a leukocyte if I come in contact with cytokines and interleukins. If I come in contact with what? I will become a platelet. Thrombopoietin, yes? So different chemicals, different hormones will cause me to be whoever. Cytokines? It's a group of chemicals, because remember, there's 5 different ones. I'm not going to make you remember each one. I know, that's cruel. All right. Predict the outcome of an overdose of the hormone erythropoietin. So by the time you hit second semester anatomy, I expect you to use your brain. Yes? Your brain. Because now you know how it works. 5, 4, 3, 2. Geez, you read slow. What's the answer? Answer. You don't know why? You don't know why you answered A, you just answered it. All right, erythropoietin causes the hematopoietic stem cell to change into what? Erythrocytes. A lot of them. Now, what's going to happen when I have too many red blood cells? Oxygen carrying capacity remains unchanged? No, because now I have too many red blood cells. What happens to that? It's going to increase. Red blood cell counts remain unchanged? No, because erythropoietin is going to make me have more. And blood viscosity decreases? No, it gets thick, because now I have too many what floating around the circulatory system? Too many red blood cells. All right, one more. What response would you expect after traveling to a high altitude for 2 weeks? Oh, that's not right. Wait a minute. Hit the wrong button. Five, 4, 3, 2. What's the what's the answer? The answer is C. Do you know why? Correct. So blood levels of oxygen will remain depressed for the duration? No. You're going to adjust. Your body's going to figure it out, right? I need more red blood cells. Yes? Yes?
Yes.
Good. All right, kiddos. Now, let's go on to the next chapter. Any questions on chapter, whatever the heck it is? Chapter 17. Exam, just to review. Tuesday, in class, multiple choice essays. What do you need to pay close attention to? Maybe this afternoonish. You need to pay close attention because I might send you a little present in the mail. And I hate it when people reject my presents. Please note, this is not your entire test. Yes? The essay questions are only a small part of your entire exam. So don't just study the essay questions and come into class and go, I thought that's all that was on the exam. No. All the stuff we talked about in class is on the exam. Question? I'm looking for a full-out answer to the question I ask you. So if I say to you, list such and such, then you can give me a list. But if I say to you, describe such and such, don't give me a list, because you can't describe something in list format. So when people say, I don't get how to answer the question, I do not want verbatim from the textbook. If I was up here and I asked you the question, I would expect you to answer it in a few sentences, right? or however long it takes to answer my question. I wouldn't expect you to get up and start reading from the textbook, right? Yeah. So I don't want that. Don't bleh all the stuff that's in the textbook. Answer the question. Does that help? So if you would, again, when you think about answering a question, essay question, how would you answer it so you could explain it to somebody else? Don't put things like it and the to describe something. Because if I was to answer your question like that, you would be really angry with me, wouldn't you? So pretend you are explaining something to somebody that does not understand what this is. And that's how you should answer a question.
Doing like the practice quizzes and the review questions in the back of each chapter, is that going to cause?
I should think it would. Are those questions going to be on your exam? No.
Well, that's not interesting.
Okay. No. But that information is going to be on your exam. And so those questions in a different way are going to be on your exam. But no, I don't use test banks and I don't use those questions. Nope. I make them all from my tiny little head. And the reason you don't get to take your tests back is because of that. Yes? What were the categories? So you need to know the hormone, who makes it, what its target is. If I have too much of it or I have too little of it, what's the matter? How was that bad? Yeah. Yeah, but if you make yourself a little chart, you can narrow down the information. What you should not be doing the night before the exam is reading this. Right? Because if you go to read this, your brain's going to blow up. Okay? What should you be studying from? Something you've created for yourself that takes all of this massive amount of information and brings it down small. So the night before exam, you should have probably 2 or 3 pieces of paper in your hand. Yes?
2 or 3?
Yeah.
My note cards like.
Yeah, you're the note card thing. But if you took the note cards and you put them out onto a piece of paper, it would be 2 or 3 pages. You have the note. You're the flashcard person. Yeah. You too. Yeah. My trouble with the flashcard thing is by the time I finished making out the flashcards, I didn't have any time to study. So I had a big pile of paper and.
When I make my flashcards, when I'm writing them,
Yeah.
that's
Yep.
how
But see, I didn't do it right.
I made about 20 of them, and then I go through stuff.
Yep.
And I make another one.
See, I was better with the pieces of paper.
That's all 3. That's all 3 of them. That's just.
And you think that's just only 2 pages?
If you took each of those and wrote down a sentence and a thing, yeah, probably about 3-4 pages. So you should be doing, and this is the reason why, and I've gotten some whiners out there, I'm not going to mention anybody, and this is the reason why Maria says 10 questions multiple choice quiz. Why? Because you should be making the flashcards as you go, as you study. You shouldn't be doing them the night before the exam. If you haven't done that, you got a few days. Spend the rest of your days studying for this exam. I'm not kidding. Because you should have been doing this all along. The average person, and I say the average person, which I am an average person, you can't get all of this in your brain in one night. No, not going to work. So that's the reason why I do these little things. You have the videos, so you can go back and create those flashcards as we go, or create notes. So many people nowadays, the young ones, they don't take notes. Why? Why do you not take notes? Okay, but you have to create something for yourself to get that information small. So if you don't take them in class, hopefully you go home and create something for yourself. We learned that last semester when we learned the nervous system. I can't handle all of this stuff. Make it small, and that's how you're going to remember it.
How many questions about?
I like even numbers, 100-ish. I like that.
Do we have a time limit or can we just?
Class time.
Oh, okay, so when we're done, we just.
Yep.
How many essays?
I don't know. They're created spontaneously. I have all the answers. You have all the answers because you've been studying. So here's the thing, guys. Not all, I'll give you a whole bunch of essay questions. Not all of them are going to be on the exam, but you have to know all of them. A, because you don't know which ones I'm going to pick. And B, because even if it's not maybe in an essay form, it's going to be in one of your questions. So you should know all this stuff as you're studying. Okay, and I'm mean.
No, you just make us learn.
I make you learn. Huh?
Do you have more than one essay?
Absolutely. Yes, several.
Yeah, it could be 8 of them on there, just simply people.
Study them all. And that's what I mean by don't throw my present away. Because when you pass in your exam, you're going to have a multiple choice section and you're going to have an essay section. And it makes me ever so sad when the essay paper is empty. And I think, you didn't at least study some of it? Some of it? A little bit of it? Yeah. So don't make me sad. Okay? The other thing I want to point out, we're hitting any other specific questions about like information and not why I'm so awful mean and give you exams. The other really important tool that you should dig out again is going to happen when you hit the cardiovascular system. What is that important tool? Interactive physiology. Does everybody know what that is? Okay, it's awesome. And this semester, it's awesome. So it's the disc you got with your book, but when you sign on to Mastering and you go into the textbook part of the book, it's right there on the left-hand column as well. So you don't need to carry this CD around with you. You can go online at any time and log into Mastering and find it as well. And hopefully you look around and see what else Mastering has to offer you, because there's so many things in there to help you study. So when we hit the cardiovascular system, be sure to check out Mastering. We're going to run a little bit of it in class when we talk about blood pressure and how to take a blood pressure. Part of the cardiovascular modules in Mastering is gives you a demonstration on how to take blood pressure, and it's really neat. So I'll run that in class when we hit that point. So those of you who are still holding out and not using it, shame on you. Awesome study tool. Again, it's that study tool you use when you are so sick of reading the book and the lines, you're reading the same line over and over again. You can pop in Mastering and listen. So when we talk about the cardiovascular system, this unit gets 2 chapters because part of the cardiovascular system is the pump. And the other part of the cardiovascular system is what? All of the tubes. That circulate what? Blood, which we just talked about. Yes? So when we talk about the cardiovascular system, we're talking about the tissues and organs and system that transport that blood. And we know what blood is for now, right? It's going to carry nutrients in the plasma. It's going to carry oxygen in some of the cells. It's going to transport those guys that protect me from foreign invaders and get them to where they need to go. And then it's also going to transport wastes from metabolism. Wastes like carbon dioxide and bilirubin and urea to the places who are going to get rid of it for me, like the kidneys and the respiratory system. That's going to help me get rid of waste too. Did you know that? Ready? Just did it. So that's how I get rid of carbon dioxide. Who's going to carry the carbon dioxide there? Cardiovascular system. Okay? So these are the transport means in which we transport blood through the body. The pump itself is a two-sided pump. Half the pump is responsible for taking blood and sending it to the lungs. And then the other half of the pump is going to take blood that comes from the lungs and send it to the rest of the system. So when you look in the book, the first thing they talk about is the pulmonary and systemic circuits. So when we look at the pump, and remember when I look at a diagram in the book, I look at it head on like this, but I have to flip myself around and lay in the book. Yes? So this is what? Right, and this is left. So the right side of the heart is going to receive blood that has already gone throughout the entire system, done its thing, transported stuff, dropped off stuff, picked up waste. With respect to respiratory gases, the right side of the heart is going to receive blood that has already been transported, dropped off oxygen, and picked up what? Carbon dioxide waste. There's 4 chambers in the heart, 2 on the right, 2 on the left. The top chambers will always get blood that's coming to the heart. They call them the receiving chambers, or the atria, or atrium, or sometimes you'll see the term auricle. That has to do with a portion of the atrium. So the 2 atrium on the top, on both sides, are receiving chambers. Blood coming back to the heart are going to always enter in the atrium. The 2 bottom chambers are ventricles. Those guys are going to push whatever's in there out. The right side of the heart is blue. How come it's blue? It's deoxygenated blood. Oxyhemoglobin, no. Deoxyhemoglobin, yes. So that's why it's blue. Is your blood really blue? No. Yeah. When you look at deoxygenated blood, it's more like the maroon color on your chairs. When you look at oxygenated blood from an artery, it's just brighter red. So why, what, huh? Yeah. Now, why does it look blue in my, like I said, well, how come the veins look blue here? Yeah, it's the color of the pigments that surround those vessels. If you go to do a dissection of a human, they're not blue and red. They're still tissue colored. They don't look, yeah. So, the blue in your diagrams indicates deoxygenated blood. So when blood goes through the system, it's going to reach or come back to the heart. And why does it have to come back to the heart? Yeah, first of all, pressure in the system is going to be really, really low. So in order for me to keep moving through all of these tubes, I need another push. Who's going to give me that push? The heart, the pump. The other reason I go back to the heart is because I need to go drop off my waste products. In the case of respiratory gases, it's carbon dioxide. And I need to pick up some new oxygen on my hemoglobin molecules. So the right side of the heart is going to receive blood from the system, and then it's going to push blood to where? The lungs. What's going to happen in the lungs? Gas exchange. So I'm going to drop off that carbon dioxide. I'm going to pick up oxygen, and look what happens to the tubes. After I pass through the lungs, and when we talk about capillary beds, we'll see this is where exchange takes place in really thin tubes. I'm going to pick up oxygen, and now my blood is what again? It's red. Oxygenated. So I'm going to go back to the heart. So what can happen? A huge push. And I need that pressure push so I can circulate to the rest of what? to the body, the system. So I got to go from here all the way to the toes, and then wrapped up in the same bunch of tubes, I got to come all the way back. So I need a good size push. So the reason we have the heart is to help push things where they need to go. The right side of the heart is called the pulmonary circuit, or the pulmonary pump. The left side of the heart is called the what? Systemic pump, or systemic circuit. So, first of all, how big is your heart? Yeah, go like this. It's about that big. And where does it sit? In the mediastinum. You remember where the mediastinum is, don't you? Between what? Between the lungs. So the mediastinum hanging out in the middle of what major? Well, it's not really a major body cavity. It's a division of a major body cavity. The thoracic cavity, yes? What major body cavity is it? The ventral body cavity, yes? Remember we broke that down into a whole bunch of little packages. The abdominal pelvic and all of those 9 little abdominal pelvic regions, yes? So above the diaphragm, thoracic. Then we have pleural on either side and mediastinum that hangs out in the middle. And that's where I'm going to find the heart. Right behind what? The sternum. What a good place. Yes, pretty well protected, isn't it? Huh? Nice protection. So we see that the heart, about this size, again 250 to 350 grams, give or take, depending on how big you are. Bigger people have bigger hearts. Is located in the mediastinum. When we talk about the heart, a little bit of a weirdness, the bottom of the heart is called the apex. Don't know why. Somebody got confused. Usually apex is top, but not when we talk about the heart. Apex is at the bottom. And what do we call the top? The base. Yeah, weird. I don't know. Somebody was standing on their head. I don't know. But the apex of the heart kind of points a little bit where? Remember, I'm in to the left. For that reason, our left lung is a little bit smaller than our right lung. And then we have major tubes that leave and come back to the heart. Because the heart sits in the mediastinum and moves constantly, hopefully, it needs protection. So the next thing, and when we dissect hearts, you're going to see there's quite a bit of fat surrounding. Yeah. After your first lab exam, we'll do heart dissection. Sheep. Bad. Bad for the sheep. Fats for what? This is a big muscle, by the way. Energy. Because I'm constantly beating whether you're sleeping or eating, right? And muscle likes to use energy from fat metabolism just as much as it likes to use energy from carbohydrate metabolism. We'll learn that in this semester as well. So fats for nutrients, constant supply, also for cushioning and protection because I'm always on the move. The other thing we have to protect us is the sternum and the ribs. And the other thing we have to protect us is membranes and fluid. So when we talk about a lot of our systems as we go along, we're also going to talk about, anatomy-wise, the membranes that surround these organs and the fluid associated with protecting these organs. So just like in your car, why do you put oil in your car? Because your husband will get really mad at you if you don't. But why do you do that? Take your two hands, put them together. heat, right? Okay, so if you don't put oil in your car, and the oil's going to create a cushion between the piston and the engine block. I hope I'm saying this right. Yeah. And the pistons go up and down to move your car. Yes? If you don't put oil in your car, what's going to happen? And you go nowhere. Yes? Because it'll get so hot, they'll fuse together. Well, that's what's going to happen in your thoracic cavity, in your mediastinum specifically, if I keep moving around in there without something to protect me, to decrease friction. So when we talk about the serous membranes associated with the cardiovascular system, doesn't that sound intelligent? We talk about a membrane called the pericardium that surrounds the heart. What do you think we're going to talk about when we talk about lungs? Pleural. Okay. So, is there fluid associated with the pericardium? What kind of fluid? It's serous fluid, but let's give it a name. We gave the membrane a name. Why don't we give the fluid a name? What was the membrane's name? Pericardium. So what do you think the fluid's name is? Pericardial fluid. Yeah. See, it's not so hard. So when we take a close look at the pericardium, it's got a couple of different layers. And the layers are fibrous, parietal. What's parietal versus visceral? These you should know. Visceral sits on the organ, parietal lines the cavity. So part of that parietal pericardium, parietal layer is fibrous and serous. So tough, protection, hold me in place covering, and serous make my what? Fluid. The fluid is to decrease friction, and we find it in the pericardial what? Cavity. The other part is something called the epicardium, and that is visceral. And what's visceral mean? Sits right on the muscle itself. So this pink stuff in here is the myocardium, and that is the heart muscle itself. Lining the inside of this pump, remember the 4 chambers? We also have a very slick, slippery surface. That's called the endocardium. It's kind of like a whiteboard. I wish I had the whiteboard. Nice and slick. You can slide right across it. That's good for blood flying into those chambers because they can slide right across the endocardium. Yes? One of the problems with making an artificial heart was not being able to replicate the smoothness of the endocardium. So when the blood came into the artificial heart, what happened? Instead of coming in and sliding in, it would bang up against the wall and the blood cells would burst or hemolyze. Yes? So that was the biggest problem for many, many years because we couldn't artificially replicate that endocardium. And the blood that was coming into the pump was being damaged and destroyed. Yeah, but still, it's not the best. It's not the best, but it's much better than it was in the beginning stages of an artificial heart. So the heart wall then is made up of the visceral layer of that serous pericardium, the myocardium, and the endocardium. And that is then going to line the heart chambers. And chambers are basically just spaces. In order for me, after we discussed muscles, in order for me to do this, what do I have to have in my hand with respect to muscles? Think about the numbers and types of muscles. Remember, if I just take a muscle, skeletal muscle cells, line them all up, what kind of movement can I do? That's it. But what if I take the muscle and put it in all different directions? What can I do? I can move them. So cardiac muscle bundles go in all different directions. And when we look at cardiac muscle cells, we're not going to see the straight line telephone poles like we saw in skeletal muscle. We're going to see cells that branch so that we can create these movements, that pump-like movement in the heart. So cardiac muscle bundles go in many different directions so we can create that pumping action. So the next thing the book talks about is chambers associated with the heart and great vessels. Great vessels, big tubes coming in and out. The atria are receiving chambers. They're at the top. And the ventricles are discharging chambers. They're at the bottom. When we dissect the actual heart, it's not going to look anything like these diagrams. And I'll tell you why. Because the diagrams give you the impression that your atria are these nice, big, round, circle things, and then the ventricles are the little dinky things, and that's not the case. When you pick up a heart, guess what most of it is? Left ventricle. Huge muscle around the left ventricle. Why do you think that's the case? Yeah, that's going to give the biggest push, strongest muscular push to all the blood that has to go throughout your system. So it's not going to look like these diagrams, and that's the reason why we do dissections in lab. So you can get a better understanding of what the tissue, what the organ actually looks like. So we see a whole bunch of diagrams, and the best one, my favorite one to see everything that we need to see, is on page 665. And that's that guy there. And he's the frontal section. So what we do is we take the heart and we face it anatomically towards you, yes? So the anterior is here and the posterior is here. And where do we make a cut? Frontal, yes? And we open it up and we look inside. In lab, you better make your frontal cuts so you can see this. Is it going to look like this exactly? No, it's going to look much different. Because when you look at the atria, they're going to be a lot smaller and much less muscular. They're going to kind of collapse on themselves. But in this diagram, we see the two receiving chambers, the two atria on either side, right and left. And then we see the two ventricles, kind of triangular shaped, right and left. And then we see the myocardium, very, very thick around the ventricles, versus the myocardium, which I don't think is marked on your diagram, that's around the atrium. And then we have all these tubes feeding in, and then we have valves. What are the valves for? Yeah, we want everything to go in one direction. The cardiovascular system is a closed system, and we want everything moving forward. When I say closed system, what does that mean? Yeah, can't go back. So my little, by the way, did the Celtics win last night?
Yes.
Phew, thank God. Got too lazy, went to sleep. Yeah, I felt safe with a 20-point lead. All right, so this little Celtics thing is closed. So say I put a pump up here. It would go around in a circle and all the way back. There's no open ends. When we talk about the lymphatic system, that's an open system. There's an end and there's a beginning. And there's an open end, but not in the cardiovascular system. So it's a series of tubes, arteries and veins, and then they are connected by very, very small tubes called capillaries. So they're never ending. So we don't want any backflow. So the valves are going to prevent backflow. There's one thing, and I know your test is coming up on the other stuff, but I just want to go through this before I leave you today. And this is kind of a hint for your next exam's essay questions. What you're going to have to make sure you're able to do is trace a drop of blood through this circuit. Tell me where you go. If you're a drop of blood and you're going through the body, what route would you take with respect to the heart? So let me just go over that real quickly. So blood goes through the system. Let's start there, and that's where I always start with my question. You've gone to the kidneys, and you've gone to the toes, and you've gone to the eyes, and you've got to come back to the heart so you can get rid of your waste, and you can get a big push to do it all over again. And blood is going to come back to the heart and always be sent to which chambers? The receiving chambers. When blood comes back from the system, they're going to come back to the heart in veins. Veins carry blood what? Back to the heart. Arteries carry blood away from the heart. Don't think oxygenated and deoxygenated because you'll get it wrong. Veins to the heart, arteries away from the heart. And we'll explain that in a second. So if I'm coming from the legs and the abdominal pelvic cavity, I'm coming this way from the bottom through the inferior vena cava. If I'm coming from the head and the shoulders, I'm coming above. What's the name of the vessel that's going to carry me back to the heart? Superior vena cava. Both of them are veins, and they carry blood back to the heart. And these guys are going to carry blood back to which side of the heart? The right side. Which part of the heart? The right atria. So blood's going to come back, and you can see where it dumps into the right atria. The next place I need to go, my little drop of blood needs to go, is I need a push. And I need a push to... Yeah, I need a push to the lungs. So who's going to help push me out of the heart? The right ventricle. So I need to go through a trap door, and that trap door is called the tricuspid valve. It's also got another name. It's called the right atrioventricular valve. Because why do you think we call it the atrioventricular valve? Because it's between the atria and the ventricles. So I push through that trap door and I go into the right ventricle. Then when the right ventricle pushes me out, I have to go through another trap door. What's the name of that trap door? It's called the semilunar valve, and the reason they call it a semilunar valve is because it looks like little half moons. And that one's going to the lungs, so we call it the pulmonary semilunar valve. Now blood is leaving the heart, so it's going to always leave out a what? It's going to leave out an artery, and since it's going to the lungs, we call it the pulmonary artery. It's blue! And the only vein that carries oxygenated blood, guess what that's going to be? Pulmonary vein. So, pulmonary artery, after I pass through the pulmonary semilunar valve, is going to take blood. How come there's two branches up there? I got two lungs. How convenient. So I'll go get my oxygen, drop off my carbon dioxide, then where do I have to go? I have to go back to the heart. How come? I need a big push this time. So I'm going to go back to the heart, always enter into a what chamber? Always an atria, except this time I'm carrying oxygen. I don't want to mix the two. So we have this interventricular septum, and there's a wall between the two atria called the interatrial septum that's going to keep the two sides separate. So blood will come always back to the heart through a what? A vein. What vein? Pulmonary vein coming from the lungs. Notice it's red into the left atria. Then I have to push through another trap door, another atrioventricular valve. This guy's got three names. There you go. It's called the left atrioventricular valve, or it's a bicuspid valve. And the tricuspid valve has three flaps. Guess how many the bicuspid valve has? Two flaps. Another name for it is the mitral valve. So I have to push through that trap door into the left ventricle. It's important to make sure you state whether it's left or right. because I have to know where I belong. So I'm in the left ventricle now, and then what's going to happen? Big push, out another semilunar valve, and this one's kind of hard to see. It's called the aortic semilunar valve. And we're going to leave with a huge push from the left ventricle out the major artery that leaves the heart to go to the system called the aorta. So that's going to be one of your questions on your next exam. Just a heads up. So we'll pick up with the valves a week from today because, of course, you know you've got your first exam on Tuesday. By the way, for those of you who think that you're going to fake illness, I will not accept anything except a doctor's note for you being absent on Tuesday.